okay? So we're also going to look at this in the context of the entire cell cycle as well. What you studied in lab was just the division part, right? How I go from one cell to two. Today we're going to look at it in the entire context of the life cycle of a cell as it begins and then at the end when it divides. So we cover cell division now because a critical part of cell division we learned in the last unit, which was DNA replication. And so we learned about DNA replication because that has to happen before division can take place, all right? So one of the reasons why we have to be able to do this, right, is that each cell we have, it needs a complete copy of DNA. And we know that because DNA has all the instructions in it that the cell needs in order to survive. And so before I go to divide a cell and make more cells, I need to make sure that each cell has a complete copy of DNA before I do that. Okay, so it's really important that we are able to take the piece that we learned from DNA synthesis and bring this back with us into this unit as well. We also know from way back at the beginning of class, which I'm sure maybe we've all forgotten about, which is that the ability to reproduce is a characteristic of living things. And that cells have to be able to reproduce. We do not form cells from scratch each time we need a new one. So cells come from other cells. So that's what that is. That's a characteristic of life, right? So when prokaryotic cells divide, and let's remember some things about prokaryotic cells. So how do we distinguish prokaryotes from eukaryotes? These don't have a nucleus, but they do have no nucleus, but they still have DNA, right? So they have no nucleus, but they still have DNA. They're all unicellular. So how many cells in an organism? Just one. And there are things like bacteria and archaea. When they divide, what are they really doing? Since they're unicellular organisms, what are they really doing when they divide? There's only one of them. So each time they divide, what are they actually doing? This is how they reproduce, right? So I have one bacteria, it divides, and now I have two. They divide, and then I have four, and so on and so forth. So for unicellular organisms, it's more akin to reproduction. This is how I get more cells. Eukaryotic organisms are a little bit different, right? So how do eukaryotes differ from prokaryotes? Well, prokaryotes didn't have a nucleus. How about eukaryotes? They do have a nucleus. Do they have DNA? Yeah, everyone's got DNA, right? They can be unicellular, but a lot of them are multicellular. And there are lots of things, but the one we're going to take a look at today are animals and plants. Okay? So why do multicellular organisms divide then? And it's a little bit different, right, than why unicellulars divide. So if I'm a multicellular organism and now I have more cells, that's not really for reproduction, right? We have a separate process for reproduction. So what would a multicellular organism do with extra cells? Well, you guys start off as one cell, and you're very, very small, but by the time you're born, you're trillions of cells because it allows you to grow. All right, so growth is one thing. I need more cells, right? Remember, they don't get bigger. That's the surface area to volume ratio issue. I can't make my cells bigger. I just need to get more of them in order to grow. All right, what else? Your skin is doing it all the time. Your hair is doing it all the time. Your liver does it all the time as well. We're shedding them, yes, but most of you aren't bald, right? And most of you have skin still because when I have more cells, I can replace and repair. So I can replace worn out cells or I can repair damaged cells. So we use this for growth. We use it for replacement and repair. When you have a cut, we repair it by having more cells replicating, right? I replace worn out red blood cells all the time. They've got about a 60 to 90 day lifespan depending on what they're doing, right? I have to replace them each time. So all of that I can use cell division for, okay? So what we know about the outcomes for cell division, whether it's unicellular or whether it's multicellular, when you divide a cell because you're replicating the DNA exactly, what is the outcome going to be? Whether you're eukaryotic or prokaryotic. So we're copying the DNA precisely and then dividing the cell into two. So what's your outcome? Do you get a new cell or do you get a cell that's identical to the one you started with? You should get an identical copy. Okay, and that's important because eukaryotes will have another mechanism later on we'll learn about called meiosis, and the outcome will be not an identical copy. Okay, so whether we're prokaryote or eukaryote, when we divide cells, the outcome will be a second cell that is an identical copy of the first. All right, so prokaryotic cells are doing this for reproduction. What does that mean about all of the offspring for bacteria? We call them daughter cells. So how do we compare the daughter cells to the parent cell? If this is for reproduction, we're using cell division, right? And we said the outcome of cell division is that you get an identical copy, okay? So this is cell division with an identical copy. So what does that let us know about the parent cell that we begin with and the daughter cells that result? They should be identical copies or the same. So the daughter cells are the same as the parent. And that's a unique type of reproduction, right? Because when we think about eukaryotic organisms reproducing, are the offspring the same as the parents? They're a combination, right, of the genetic information from the parent. So this is unique to prokaryotic organisms, and it's unique to them because they're the only ones that use cell division for reproduction. So we're not going to use it for reproduction, and that's how we're going to get offspring that are not exactly the same, okay? But if we loosely look at how this works, it's slightly more complicated than this figure leads you to believe, but that's okay. We begin with an origin of replication in the DNA. Do you guys remember what happens at the origin of replication? What do we do there? This is where you just took this exam. You open your replication fork. I either have good or bad news for you depending on how you look at these things. You do know that the final exam for this class is cumulative. So you cannot have deleted everything from Unit 3 because it's going to come back again for you in the final exam. So the origin of replication, just like your origin of replication, this is where we open the replication fork to do what? We're going to copy yes, this is where we open the replication fork to copy DNA. Because on the previous slide, we said we had to learn about DNA synthesis first because prior to cell division, we have to copy the DNA. And again, this doesn't matter if you're prokaryotic or eukaryotic, okay? So you begin with a cell that has one set of DNA, and when you end, what do you have? You have two copies, right? So in the parent cell, this is the one we started with, you have one chromosome. After replication, how many do I have? After replication, I have two chromosomes. Take a look, has my cell split yet? I only see one there, right? So there's a brief period of time where this cell has twice as much DNA as it needs, okay? Because it has two chromosomes instead of one. After the copying of the DNA is complete, then we begin the actual process of splitting the cells. When you split the cells, remember we have two chromosomes, right? But I want to end up where each cell has just one, because that's how we started, right? So I want the daughter cells to end up exactly like the parent cells, which means the daughter cells each have to have one chromosome. So I'm going to take the 2 chromosomes and I'm going to split the cells, and when we're done, you have 2 daughter cells, and how many chromosomes are in each of the daughter cells? You have 1. So there's 1 chromosome in one daughter cell, 1 chromosome in the other. Okay? So there'll just be a brief period of time, though, where your parent cell actually has twice as much DNA right before it splits. For eukaryotes, it's a little more complex than that. So for the eukaryotes, we take a step back and we look at it in the context of what we call the cell cycle. So the cell cycle is the life of a cell, and it begins right after the cell divides. So right after the cell divides, this is where we start. This is where we have a newly formed daughter cell. It's always somebody's daughter cell, right? Because cells have to come from other cells. So when the life cycle starts again with a newly formed daughter cell, it's because a division has taken place. So we enter into the first part of the cell cycle, which is called the gap 1 phase. We also call it G1, and this is part of a larger phase that we call interphase. So G1 is where you have normal function taking place. So in this place, this is where like liver cells would function as liver cells, and kidney cells function as kidney cells, and whatever your cell is supposed to do normally, that's what it's doing during the G1 phase, okay? Upon a signal, we get ready for replication. So we're gonna get ready to divide by entering into the next phase. This is still part of interphase, but now we have a part of interphase that we call the S phase. S stands for synthesis because we're gonna replicate our DNA. This is DNA synthesis, right? So in the prior example we looked at, we had 1 chromosome, and then after the S phase was over, it had 2 chromosomes. Well, most eukaryotic cells have more than 1 chromosome. So at the end of the S phase, you're going to have double the amount of genetic information. And just so you know, for our cells, we get a number on them. We call them 2N, and that N can be anything. That N is specific to the cell. It's actually specific to the species. But in interphase in G1, the amount of DNA that you have is called 2N. So after the S phase is over, what are you going to have if it's double the amount of DNA? What's 2 times 2, friends? 4N. So you'll have 4N after the S phase. You're then going to enter into something called G2. During G2, we're going to get ready for division. This is also part of interphase. So in the gap 2 phase, we prepare for cell division. And how much DNA does the cell have at this point? So during the gap 2 phase, while we are preparing for cell division, did I say anything about additional DNA replication? Yes, no? Like look at does it say anything about additional DNA replication there? Okay, so if we had 4N when we started the G2 phase, but I haven't touched the DNA at all, what do I have at the end of G2? I still have 4N, nothing's changed, right? All right, I head into mitosis, which you're already aware has 4 stages. You learned about prophase, right? Metaphase, anaphase, and telophase, during those phases, I still have 4N. It's only at the end when we complete mitosis and we do something called cytokinesis, where I split my cells in two. After cytokinesis, how much DNA do we have? So remember, we're going to split it in two. Remember, we saw in the prokaryotic cell, I had one chromosome. We ended up with 2 chromosomes, and then I split it, and each daughter cell had 1 chromosome. So if I have 4N through mitosis, when I am done with cytokinesis, what do we return back to? What's 4 divided by 2? We're going to go back to 2N, okay? And that's exactly where we want to end, because that's exactly where we started, and we want to end and start in the same place because the daughter cells should be identical to the parent cell that we started with, okay? After cytokinesis, I have 2 cells, right? And they are both 2N, okay? And they should be identical to the parent. And then both of those daughter cells will start over again, right? Because that's the newly formed daughter cells. They restart in interphase G1, and they go on to be their own cell. There is one exception, the little exception that you see off to the side there. This is called gap 0. This is for cells that do not divide. So you'll notice G0 just stays inside as part of G1, and we never get to the S phase, which means we never replicate our DNA, which means we don't undergo mitosis, okay? So cells that do not divide, there are 2 major examples in the human body. The first are cardiac myocytes. What do they make up? That is the heart muscle cells. And certain types of neurons. Where do you find neurons? In your brain and your spinal cord. After a period of time, those cells stop replicating. So neurons usually stop replicating earlier than cardiac myocytes. Neurons usually stop dividing in utero. So you're born with all of the neurons in most parts of your brain that you will have for the entirety of your life, because they enter into gap 0 and then they do not divide. So that means that they don't divide, which means they cannot repair themselves. We always make the joke about doing brain damage, but brain damage is permanent because there is no way to get those cells to replicate and repair themselves or replace themselves. The same is true for cardiac myocytes, though they stop replication usually right before you enter into puberty. So around 10 or 11 years old, your cardiac myocytes will stop dividing. So at that age, your heart is the biggest it will be for the rest of your life. This is also why we worry about people that have heart attacks, because when they damage the muscle of their neuron, of their heart, right, those cells cannot divide and repair themselves. So when you have a heart attack, you're left with scar tissue, no longer functioning cardiac myocytes. You cannot replace them. So a part of your heart will always be non-functioning scar tissue. And that's why we want you to take care of them, because you cannot fix them once they are broken, and you cannot replace them once they are lost. Everybody else, though, they're fine. They'll keep undergoing cell cycle for a period of time. Okay? So one of the ways that we identify what type of phase our cells are in is by looking at them at the cellular level, okay? So when I look at cells that are in interphase, when I look at them when they are in S and G2, at the cellular level, what did you guys look for to let you know that you were looking at something in interphase, in lab? What were you looking for? So there's your nucleus, right? Here's your plasma membrane. Based on the shape of the cell, by the way, is this an animal cell or a plant cell? This is animal. What was the distinguishing feature, though, that you looked for? Well, okay, that'll tell me that it's an animal cell, because there is no cell wall. Sorry. What's the feature for interphase? I look at the nucleus and... Did you see anything specific? You had a lot of diffuse staining. That's how I knew I was looking at the nucleus, but no chromosomes, right? Because at this point, at the molecular level, right? So this is cellular. That's what I can see in the microscope. At the molecular level, your DNA is sitting in a chromatin format. Do you guys remember all the way back from Unit 1 what chromatin is? So do you know what a chromosome is? In chromosomes, they're tightly wound up. So chromatin is the opposite of that. So these are this loosely wound. It's very stringy. And this lets us access, or it lets the cell access, the information that's in the DNA, which we're going to learn about on Wednesday. So I don't want to bind up the DNA really tightly because I want to be able to read the information that's in it during G1, because this is where I'm going to be doing a lot of protein synthesis, and this is where we're going to have growth and things happening. So I want to be able to get to the information, all right? So to do that, we're going to find the DNA in its chromatin format, and that's why I don't see a whole lot of distinct structures in the nucleus right now, because they aren't present yet. I haven't wound the DNA up very tightly because I don't want to just yet, okay? So if everything is okay, the cell will get a signal to begin the next phase. So we'll signal the continuation of the cell cycle. And after G1, where do we go? G1 goes to S phase, okay? We're still in the S phase, but now this is where we're going to start to see the formation of chromosomes, okay? So during the S phase, remember we said the S stands for DNA synthesis. I want to go ahead and copy those pieces of DNA. Can I copy the DNA if they're tightly wound in chromosomes? Not yet, okay? So I'm still going to be in my chromatin format. So at the cellular level, I should still see chromatin. The pictures don't change much for interphase. I'm still going to see chromatin. I will still have a distinct nucleus. You'll be able to see the nucleus, correct? That won't go away yet. At a molecular level, though, we are replicating the DNA. So the origins of replication open. We get replication forks until we completely copy the DNA from one end to the other, okay? The pieces of DNA that are being copied are referred to as the genome. Do you guys remember, or maybe you know what a genome is? Have you heard the term before? We're always big on sequencing the human genome. That huge piece of DNA that you have, that's your genome, right? And because it's DNA, really what we're telling you is that it's all of the instructions that your cells need. So the genome is what is inside of the DNA. All of the instructions to be a human cell are in the human genome. In order to make it a little bit easier for us to work with, we talk about them in their chromosome format. So as we get ready to divide our cells, we're going to package them up into chromosomes, okay? You have one big piece of chromatin. One piece of chromatin makes up your genome. That's all of the instructions possible. Later on, though, we are going to break up that one piece of chromatin into smaller packaged chromosomes. So I take the one big piece of chromatin and I'm going to break it down into chromosomes, okay? You'll have 46 of those. So I take one gigantic piece, break it into 46, okay? All right, let's test our skills from the cell cycle. As we enter into the S phase with 46 chromosomes, we said that was equal to 2N, right? When I have finished the S phase, what do I have when we're done? So we start with 46. What do we end with? We end with 92, and 92 is equal to what? 92 is equal to 4N, okay? Because I had 46 to start with, right? And what do I do during the S phase? I make a complete copy, right? So if a complete copy is 46, I end up with, yeah. All right, so now you have 92 chromosomes, we're in 4N, but they're still actually in their chromatin format because if you remember, you couldn't distinguish between the G1 phase, the S phase, and the G2 phase, right? We just told you to call everything that was indistinct. We told you to just call it interphase because at the cellular level, they all look the same, okay? So the chromosome number that you have varies amongst organisms, right? I told you the value of N was specific to the species. And most eukaryotes range anywhere from 10 to 50 chromosomes, okay? What we're talking about right now is another vocabulary word that we refer to as somatic cells. We want to hold on to that one because somatic cells will be found in both males and females. And a better way to think of this, a better definition, is just to say that somatic cells are not used for reproduction. So all of the cells in your body, except for the ones that are used for reproduction, okay, are considered somatic because both males and females have them. Later on when we do meiosis, that's when we'll pick up with what we call gametes. Gametes will be specific to males or females, okay? But like liver, kidney, cardiac myocytes, skin, all of those are somatic because both males and females will have them. And they are not used for reproduction, okay? So I told you that you all begin with, in G1, you begin with 2N, okay? So N is equal to the chromosome number. And in humans, Rn is equal to 23. So we have 23 different types of chromosomes. So we take our long, big piece of chromatin, break it up into 23 sections. We have 23 of them. But when we're in G1, we are not N. What are we? We're 2N. This is a really basic algebraic formula, right? So 2, what's N again? 23. What's 2 times 23? 46. That's how we get the number 46. That's how we know how many chromosomes we have. But that implies that we don't have one copy of the chromosomes. How many do we have? We have 2 of each copy. That's because one set of your chromosomes is maternal in nature. So who do you get that from? From your mom. The other copy is paternal in nature. Who do you get that from? You get that from your dad. And you are always 2N because you have a complete copy of DNA from your mother and a complete copy of DNA from your father. That's where we get 46 from. But there's actually only 23 distinct types of chromosomes. We just find them paired. One maternal with one paternal, okay? The paired chromosomes in your somatic cells are referred to as a homologous chromosome. Homologs contain the same information about the same traits, but the information can vary. a good example of this, well, actually it's a terrible example of this, is hair color. It's sort of terrible because there's more than one gene that controls it. Okay, so for example, the hair color gene is found on the same chromosome in the same place. But mom might give you a gene for brown hair, and dad might give you a gene for blonde hair. Okay? So it's the same trait, which is hair color, but the information can vary because mom can give you one version and dad can give you a different version. Okay? Because you have two of everything, we refer to your somatic cells as diploid, because what does di mean? Two. You have two of everything, right? One from mom and one from dad. So if we look at homologous chromosome, we're going to pretend that this is chromosome 14. We could have picked anything from 1 to 23, but we chose 14 here. And let's go back to our hair color idea, right? So let's put hair color here. Hair color is in the same place on chromosome 14. That's what makes them homologous to each other. But perhaps, like we said, mom gave you a gene for brown hair color, and dad gave you a gene for blonde hair color. Okay? So it's the same trait, which is the color of your hair, but it's a different version of the trait. Okay? So together, these are referred to as homologous pairs. You get one from mom, that's your maternal copy, and one from dad, which is your paternal copy. And this is considered 2N because you have this chromosome, and for each one you have 2 copies, okay? So after replication takes place, because remember, in the next cell, I want one of these purple chromosomes, but I also need one of these green chromosomes. I don't want to split these 2 chromosomes from each other because then my cell will only have a purple or a green, correct? So after synthesis, what I want to do is make sure that I've duplicated both of these chromosomes, okay? And I want those chromosomes to be duplicated exactly because in my next cell, I want each one to have a perfect copy of the purple one, right? And what version was the purple one carrying with it? For brown, right? And I want to have a perfect copy of the green one, right? And each green one has the blonde, okay? So I synthesized my DNA. So now instead of having 1 purple chromosome, I now have 2. And instead of having 1 green chromosome, I have 2, okay? Over here, we said this was 2N, right? Because I had a chromosome and there were 2 copies. How many copies do I have here? I have 4. This is where we get 4N from. So we're in 4N after the S phase, right? When your chromosomes are identical copies to each other, we call them sister chromatids, right? Sisters are identical to each other. So the sisters will remain attached to each other until we split them during mitosis. When I split the 2 purples from each other, that means that each cell will get 1 copy of the purple chromosome. And when I split the green chromosomes from each other, that means that each cell will get one green copy, right? And so you'll end up with a cell that has 1 purple and 1 green when I'm all done, okay? It is incredibly important to understand the difference between homologs and sisters. Right now, we're going to get ready to split sisters from each other. So those are identical copies to each other. But later on, you're going to have to remember that homologs get split in meiosis. So you got to be careful, okay? So you have to understand who's homologous and who's a sister. So after the DNA has been replicated, we begin the condensation phase. You're not actually going to see this really taking place until prophase, but the duplicated chromosomes where you have two sisters attached to each other. The two sisters remain attached to each other through a set of proteins called cohesins. Cohesins make sure that the two sisters stay together. I want them to stay together until right before I split them, okay? There's also an area called the centromere. So let's look at the cohesins really quickly. So the cohesins are going to be in this area here so that the two chromosomes stay attached to each other, okay? And then you have something called the centromere. The centromere is where the chromatids are most loosely attached, and that's going to be the band that goes this way, okay? So when we show you them like this, a lot of times they look like butterflies. That's the centromere, okay? We are going to attach the centriole spindle fibers to the centromeres, and it's where they are attached. That's where we're going to actually pull them apart later on during mitosis, okay? Again, this is all happening at the molecular level. You can't really see this at the cellular level, but you can see the condensation because you'll start to see what inside of the nucleus. You'll start to see chromosomes inside the nucleus. So you'll see the condensation part. You won't necessarily see the formation of the cohesins and the centromeres, at least yet. Okay? So we will just double check that we are okay, because I think maybe the hardest part of mitosis for some strange reason is learning how many chromosomes we have at any given time. This is a cell. This is a parental cell. This parental cell has a single chromosome in it. So it is clearly not human, because if it was human, how many chromosomes would it have? It would have 46. So there's not 46 there, so this is not a human cell. We're just going to use this for an example. After the S phase, the parental cell is going to duplicate the amount of DNA it has, excuse me. So it goes from having one chromosome to having two, okay? We call these two sisters because they are identical to each other. I then want to split the sisters after mitosis so that I end up with you have two daughter cells, right? The two daughter cells have the same amount of genetic information as the parental cell, and it should be identical genetic information, and that's good cell division, right? Because I should have two cells at the end that are identical to the parental cell that we started with, okay? This cell, unlike us, goes from N to 2N back down to N. Okay? For humans, we have 46 chromosomes, but remember that's our 2N number because we're going to get one set from mom and one set from dad. They're arranged into 23 pairs because there are only 23 distinct chromosomes for humans. We arrange them in this format where you have the largest with the most genetic information being number 1, all the way down to the smallest chromosome, which is chromosome 22. These are what we call autosomes, and they are identical in both sexes. The difference between the sexes then is the sex pair. The sex pair is the 23rd pair, but it's not really a pair. In females, it is a true pair because they're XX, but in males, it's not an identical pair because they're XY. But that's 23. So you have 22 autosomes in one sex pair. Okay? That's where we get the 2N from. So this implies, for example, that this version of 1 was paternal, and this version of 1 was paternal, and this version of 3 and 4 and 5 were paternal. Where did the other copy come from? So where did this copy of 1 come from? that had to be maternal, and this copy of 2 was maternal, and this one of 3 was maternal, 4 was maternal, 5 was maternal, right? So that each pair that we're looking at, we get 2 copies of the chromosomes, 1 from mom, 1 from dad, and that's how we get to 46, okay? How do we feel about chromosome numbers? We're okay? Yes, no? So we're going to enter into G2 now. We use the second gap to get ready for replication. Let's think about this first for a second. At the cellular level, what do I see? So if I think about looking into a microscope, am I going to see anything fun in the nucleus? Will I see chromosomes yet? Not really. No chromosomes yet. I'll still see a nucleus, right? That hasn't changed. And again, that's why when you looked at the microscopy things, everything was interphase if you couldn't see chromosomes, because we can't tell the phases apart at the cellular level. At the molecular level, though, we know lots of things are going on, right? So we're no longer 2N. What are we now? We are 4N because we've synthesized all of our DNA. We've duplicated it. We're sitting at 4N. We have twice as much DNA as normal, 2 times the amount of DNA. And at this point, our cell is going to get a little bit bigger. It's going to increase the amount of cytoplasm it has. It's going to replicate cell membrane. It's also going to replicate a lot of the organelles, because that's going to help the cell when it gets ready to divide and ensures that both cells can survive. So think about this. If I don't have enough mitochondria for both cells to divide, right, and they separate from each other, there's not enough mitochondria to make ATP, what happens to both of my cells then? If there's not enough mitochondria, they'll both die, right? So this phase ensures that I have enough resources and I have enough organelles so that when we go to split, I know everybody will be able to survive, okay? If everything is good, so we inventory check and we also make sure we have enough DNA, if everything is good, we will then enter into mitosis. So the first phase of mitosis is called prophase. And at the microscopic or cellular level, that's when we start to see chromosomes. The key here, though, is that although I see chromosomes starting to form, they are condensing. Are they organized in any specific way? Not yet. So there's no specific organization yet. It's coming, but not in this phase, right? So I'll see chromosomes condense for the first time, but they have no specific organization. We will begin to see the nuclear envelope degrade. So eventually there will be no nucleus visible. And the other thing we're going to start to see, though you probably can't see it at the cellular level just yet, is you will start to see the spindle form out of the centrioles. So you may remember that the centrioles are a microtubule organizing center, and the spindles will grow towards the center of the cell, because we start to think about an imaginary line down the center of the cell at this point, okay? Do plants have centrioles? They do not, but they do have spindle fibers. So no centrioles, but they do have spindles. We have to separate their tubules in some way, right? So they don't have centrioles, but they do have spindles. So we will still see a spindle formation. We just won't see them coming out of the microtubule organizing center. All right, for metaphase, our chromosomes are going to attach to the spindle at the middle of the cell. So remember, I told you we're going to draw an imaginary line down the center. What lines up at the center of your cell? The chromosomes. So I'll continue to see chromosomes, right? But now they're going to have a specific organization. You may have noted that the chromosome itself wasn't exactly down the center. What lines up perfectly down the center is the centrosome of the cell, right? So those are the things that are actually going to line up perfectly down the center of the cell. The arms and the legs of the chromosomes can lay in any shape or form we want. It's just that we want to make sure that the center of the chromosome is lined up with the center, the imaginary midline of the cell. The spindle fibers are here. This is the microtubule organizing center, the centriole. And the spindle fibers project towards the center of the cell. You also have something called astral fibers, which project away. Those are not necessary for separation of the chromosomes. We're primarily concerned with the spindle fibers, which are growing towards the center of your chromosomes. Then it's not going to divide correctly. Okay, we're good with metaphase. Metaphase, by the way, stands for middle, because the chromosomes are all lining up in the middle. All right, once you have your spindle fibers attached, they will do so in a site along the centromere called the kinetochore. Okay, so in the centromere region, the microtubules actually attach to the kinetochore, and you'll notice that we get attachments on both sides, because remember, I want to go ahead and split these sisters. One sister is going to head towards one cell. The other sister will be pulled in the opposite direction towards the other pole of the cell. And this is how we're going to split them, because remember, I want to split all of the sisters during anaphase, which is the next part. Okay? I have to make sure my spindle fibers are attached. If I ensure that they have attached, that's when you'll see anaphase. So for anaphase, we're going to reverse the microtubules. Remember, during metaphase, they were growing towards the center. Now I'm going to turn around and have them grow in the opposite direction, back towards the microtubule organizing centers. And when they grow in the opposite direction, they will pull their sister with them. Okay? So you can actually see two sets of chromosomes. There's a set that's being pulled towards this microtubule organizing center, and there's a set that's being pulled towards the opposite microtubule organizing center. Okay? You can visually see this, which I'm sure you did, right, during lab. You can see the two sets separating from each other as the spindle fibers retract back towards the poles. Okay? It is still one big cell, though, right? If you take a look at the membrane, it's still one big cell at this point, correct? So just double check us at the molecular level. What are we? Are we 2N or 4N? It's still one big cell, guys. I know they're separating from each other, but we're still, yeah, the cell is still 4N because I still have one big cell, okay? Telophase is the final part of mitosis. For this, we start to see the nuclear envelope reforming. It'll reform around each set of chromosomes. The chromosomes will start to unwind as well, right? And what are they unwinding back into? What's the other C word? Into chromatin, okay? Your spindle fibers begin to disappear. Your centrioles will migrate back, okay? And we begin to see two separate cells, but they haven't completely separated yet, correct? Do you still see there's like a gap where they're still connected to each other as we continue to pull the chromosomes away? This cell is not completely divided yet, and so this cell is still considered, we're still considered 4N because we haven't completely sealed the cell off. This is still technically one gigantic cell, okay? The final step, which is not a stage, it is the final step. This is called cytokinesis. When cytokinesis is complete, we will then have two complete cells. It happens differently depending on whether you're an animal cell or a plant cell. For animal cells, it's super easy. I'm just going to take my plasma membrane and pinch it really tight. You form something called a cleavage furrow, which is happening here. That's where the plasma membrane begins to separate. There's a set of contractile microfilaments made of actin that pinch the cell apart. Oh, you guys know what a drawstring, like drawstring pants? If you just keep pulling and pulling and pulling and pulling, eventually it'll pinch the two cells apart. And so that's what the microfilaments are like, pulling on a drawstring until it separates the two cells. Once you have completely separated the two cells, they are now considered 2N, and both of these cells enter back into what phase? These both return back into G1 of interphase, okay? But only after I have completely pinched them apart. Plant cells are gonna be a little bit different, and that's because we have something that we have to contend with, which is the cell wall. So you will still get pinching of the plasma membrane, but I also have to have formation of what we call a cell plate. You can usually see the cell plates forming during telophase, but the cell plate won't be complete until cytokinesis. And once it's complete, then you have two completely separated cells from each other. Both of these are also 2N, and both of these cells go enter into G1. We start all over again. Okay? Do you have any questions about the mechanics for mitosis? So you need to know what's happening at the cellular level for each phase. So that would be like, how do I know the cell I'm looking at is in this phase? By looking at it in a microscope. You also need to know what's happening at the molecular level, right? So what are the details that I can't see with my eye? For example, how much DNA does it have? Am I getting spindle fibers forming? Things like that, okay? Everyone's okay with all of that? And you'll be able to tell the difference between a plant and an animal cell by looking at them? Fantastic. Okay, so then let's look at this part. This is controlling our cell cycle. So cell cycles get controlled by checkpoints. Checkpoints ensure that everything will be complete as you move from one phase to the next. And it's particularly important in interphase because we want to make sure that if we are going to attempt a cell division, we can complete it successfully. I don't want to get halfway through cell division and be like, oh, I ran out of mitochondria here. Okay, because that means the cell's going to die. It won't be able to successfully complete. So I only want to undertake this if everything is good. And in order to ensure that everything is accurate, we're going to go ahead and make sure that we have these checkpoints here. Okay? So we get a lot of feedback from the cell. The cell provides its own feedback, which allows it to either advance to the next part of the cell cycle or not. Okay? One of the biggest types of feedback we get is the feedback that determines whether or not we enter into the S phase. And one of the ways that cells do this is through a process known as contact inhibition. So contact inhibition is the cells sensing how closely or tightly packed they are with another cell. If there is no room to grow or to divide, they are undergoing something known as contact inhibition. So if they're touching each other, they are inhibited. So if they're touching each other, if they touch, they won't divide. Because if they're touching their nearest neighbor, what does that mean? There's no more space. There's no more room, right? And if there's no more room, should I keep dividing? That's how you end up with cancer, okay? If there's no more room, then you should undergo contact inhibition and the cells will not exit out of G1. They'll just hang out in G1, okay? Should I go into S phase if I'm not gonna divide? No, bad idea, okay? So if we're undergoing contact inhibition, we'll just hang out in G1, okay? If some cells die, that's gonna open up some space. And if some space opens up, we will no longer be under contact inhibition and we can add more cells by allowing them to divide, okay? So that's one major checkpoint. There are also 3 other checkpoints inside of the cell cycle that we look at as well to help control the phase. So we go back and look at our phase again. All right, so let's say there's room to grow. And so my cell is going to enter into what we call the G1 checkpoint. The G1 checkpoint is inventory. So this ensures that I have enough stuff, I have enough resources. Like I don't want to get halfway through my DNA and be like, oh, I ran out of thymines. Oops, okay? So the inventory checkpoint allows us to make sure that the cell is healthy enough and it has enough resources to complete replication, okay? So if we pass the G1 checkpoint, that allows the cell to go into the S phase and replicate the DNA, right? After we're done replicating the DNA, PS, what are we? Are we N, 2N, or 4N? After we finish S phase, are we N, 2N, or 4N, humans? You are 4N, right? We're 2N over here. We're 4N at the end here. Then we enter into the G2, but there is a G2 checkpoint. The G2 checkpoint checks to make sure that DNA synthesis is complete. So it'll go through and check and make sure that you are actually 4N. Why is this an important thing? If I don't have 4N, when I go to split this cell, will I end up with the exact same amount of genetic information I started with? You'll have an incomplete copy of DNA, and that means one of your cells isn't getting what it needs to survive, okay? So if you have like 3.5N, that means that somebody's gonna get gypped when you go to separate your DNA, and you don't want that, because cells that don't have complete copies of DNA can become cancer cells. Okay, so we'll double check the DNA synthesis has been completed in the G2 phase. We'll make sure everything's 4N and it's accurate. And if it's 4N and everything's accurate, then the cell is allowed to proceed into mitosis. And then it'll move along through the checkpoint until it hits the M checkpoint. Now this M doesn't stand for mitosis, this M stands for metaphase. What's happening at metaphase, in metaphase? This is also referred to as the spindle checkpoint. But what's happening in metaphase? Everyone's lined up down the center, but also what else is happening? I need to make sure that I have spindle fibers, right, attached to everybody, correct? Because when I have the spindle fibers retract, they should each pull a sister chromatid with them, correct? Okay, so what happens then if we miss a chromosome? Let's pretend this one didn't get hooked up correctly. When you go to separate these, this cell is going to be missing this chromatid, right? So this cell is going to be -1. What happens to the other cell? Because when it retracts, it's going to end up pulling 2 chromosomes with it, right? So this cell that forms over here isn't going to be -1, it's going to be +1. Neither of these cells is completed accurately, right? This cell will have too little information, and this cell will have too much. And now I've just wasted all of this time and all of my resources because both of these cells will end up dying. Having too much information, by the way, in a cell is just as bad as not having enough. You have to have just the right amount. This is like Goldilocks. Everything has to be just right, otherwise the cell will die. Sorry, Braden, go ahead.
Is metaphase in chromatin format or chromosome format?
Chromosome format. Once you hit prophase, we're chromosomes all the way to the end. Okay? So that M, the metaphase spindle checkpoint, ensures that when I go to divide the sisters from each other, everything will be just right. Okay? Sorry. I'm sure it's incredibly important. So when we don't do this process correctly, that's when we end up with problems, okay? Classically, when we have faulty cell division, it usually causes cancer to arise. And that's because your cells aren't following the cell cycle properly. Cancer cells don't have the right amount of genetic information, and each time they replicate, the copies that they make also carry those mistakes as well. And without the proper amount of information, there's nothing to tell the cell to stop dividing. So cancer cells oftentimes don't have the checkpoints, so they just keep making mistakes. They also oftentimes don't have the contact inhibition part that goes along with it as well. So they will continue to take those mistakes and they will grow in places they're not supposed to at times that they're not supposed to. Okay? So there's a lot of things that cause cancer. Most of them, though, result from damage to the DNA. Because all of the instructions to replicate correctly are found in the DNA. So if you damage the DNA, then they can't replicate correctly, and it just creates this cycle where you end up with cells that are unable to divide appropriately. You can get external damage to your DNA. Okay? So that means it's coming from the outside. That's going to be things like UV light and cigarette smoke. Okay, all the carcinogenic chemicals that people hang out with. Those all are external factors that damage your DNA. You can also get internal or inherited factors as well. So these all come from the inside, and these are going to be things like changes to hormonal patterns and mutations, which are changes in the DNA. Remember, when DNA doesn't replicate itself accurately, we call that a mutation. At the very end of the last unit, we looked at some things, right? We looked at proofreading. We said that the DNA had to go back in and check and make sure that everything had been synthesized appropriately. If it doesn't proofread correctly and that mistake is allowed to stay in the DNA, we call that a mutation. They are bad, by the way, because that means your information is incorrect. So cancer oftentimes begins with a normal cell that has completely normal DNA, but something causes the DNA to get damaged, either one of those external factors or an internal factor. Classically, when we think about it, you get a cluster of cells called a tumor. They've lost contact inhibition, so even though they know there's no more room, they continue to grow on top of each other, and that's where you get the tumors from. Because think about it, what do you think a tumor feels like or looks like? It's a lump, right? You're looking for a lump or a mass of cells. Those cells have lost contact inhibition, so they'll continue to grow on top of each other, okay? We can get two types of tumors. There's actually lots, but for our purposes, let's pretend there's just two of them. The first are called benign tumors. A lot of you have these already. If you have a freckle, if you have sometimes they're just benign, and you know it because you've gone to a dermatologist. Those benign tumors are always surrounded by a healthy layer of cells, and so that healthy layer of cells protects the other cells from the cancerous cells. These are also referred to as encapsulated because they're surrounded, and they cannot, and here's the key, they cannot spread to other areas because they're inside a capsule of healthy cells that won't let them escape. Benign tumors are oftentimes removed easily from the body. You just surgically resect them and everything's okay. The only time they get to be complicated is when the benign tumor is in some place that's difficult to reach, like your brain, spinal cord, where you might do damage to other parts of the body while you're trying to get to the benign tumor. Malignant tumors are the ones that everybody worries about because these are not encapsulated. So there's nothing stopping them from spreading. We refer to them as invasive because they can grow beyond their borders into other tissues, okay? If they leave the primary site where they grew and go to other parts of the body, we refer to it as a metastasis. Have you guys heard of these terms before? And it's sad to say, living here in the United States, that we're talking about 1 in 5 of you getting Alzheimer's. Well, you're much more likely to either be affected by cancer or know somebody, because I think the rate right now in the US is 1 in 3 people will end up with some type of cancer in their lifetime. So it would be incredibly unusual to find somebody who either has not had some type of cancer or knows nobody in their family and friends who have had it, okay? So cancer cells that are unencapsulated grow rapidly, okay? and because they're not encapsulated, they can go to other parts of the tissue. So carcinomas, for example, begin in the lining. So this carcinoma would have begun here in the lining and the epithelium of the lung. But look, instead of growing in this nice ordered row, it grew out of control. After it grew out of control, it started to break off, and these cancerous cells, especially in the lung, it's problematic because you have a lot of blood vessels, and then these cells can go anywhere where your blood goes. And where does your blood go? Everywhere, right? So classically, we start to see brain tumors. So the two most common sites for lung cancer, brain and bone for metastases, okay? Because you can get the cells in the bloodstream. They can also go through the lymphatic vessels as well. If you know somebody who's had cancer before, a lot of times we do lymph node checks because we want to make sure that the cancer cells haven't gotten themselves into the lymph nodes and spread to other places as well. So you look for whole body scans to make sure they haven't hit the blood and gone everywhere. And you also look for the lymph nodes in the surrounding area to make sure that the cancer cells haven't gotten there either. So I want to talk to you about some intrinsic changes then as well. So these are going to be changes in the genes inside of you, okay? So there's two major genes that we look at for helping to control cell division, and they're sort of the opposite of each other. The first gene is called P53, and this is a G1 checkpoint, okay? This isn't the inventory checkpoint. This is a different checkpoint inside of G1. So what it normally does is it detects abnormal DNA. All right, so somehow we had a mistake that took place, we replicated the cell, and then P53 double checks to make sure that the cell's DNA is completely normal. If the cell has abnormal DNA, should we let it replicate again? No, okay? If you end up with like UV damage, because you were like, I'm gonna stay outside all day in the sun without sunblock because it's cloudy, but that doesn't help, and you damage your DNA through UV light, should we let those cells replicate? So P53 will stop a cell, okay? P53 stops the cell, and it usually directs the cell to destroy itself. So we call this apoptosis. This is selected cell suicide. Is it better to kill a cell that is damaged or to let it just hang out damaged? It's better to kill the cell. I want to get rid of it, okay? So P53 will direct any damaged cells to undergo suicide, kill themselves, okay? If P53 is the gene that gets damaged, though, there's nobody to tell that cell that its DNA is damaged, and that means that the cell will just continue to divide unchecked, and every time it divides, it will copy that mistake, okay? So that P53 gene is really important. This is one of the genes that we sequence. We sequence this one. Have you heard of the BRCA gene before? BRCA. That's a common one we use as an indicator for breast cancer. It's used for ovarian cancer, a couple other ones as well, okay? We look at these genes, and if those genes are damaged in any way, shape, or form, the patient who has the damaged copy runs the risk of developing cancer. So we can give you your lifetime likelihood of that happening, if that's something you're interested in. Most patients don't want to know, okay? So we class the cancer-forming genes into two things. The first are called proto-oncogenes. So proto-oncogenes, proto means that they could become oncogenes, cancer-causing genes, okay? So these genes are actually necessary for cell division. So particularly during development, when you need lots of cells, right? Right after you've been fertilized and we're trying to get you into a big embryo, during development, all of those proto-oncogenes are on. And they're on because we want them to stimulate cell division. But as you get older, what do you think happens to your proto-oncogenes? And when you don't need as much cell division because you're not growing as much, we tend to turn them off, okay? So they're no longer expressing. If you damage a proto-oncogene, though, it becomes an oncogene because it will cause cancer. So proto means it could, but it hasn't yet, and they do have normal functions. But as an adult, if you damage that proto-oncogene, it's now just an oncogene, and it will cause cancer because the cells are going to replicate uncontrollably. The other genes that get damaged are called tumor suppressors, okay? So the good way to think about this is the gas pedal because it makes replication happen faster. So if you damage the gas pedal and you just put your foot all the way down, you're just going to keep dividing and dividing and end up with cancer. Tumor suppressor genes are classically considered brakes because when you step on a tumor suppressor gene or express a tumor suppressor gene, it turns cell division off. A lot of the genes that allow the cell to know that it's undergoing contact inhibition are tumor suppressor genes. Okay, cell touches something and the tumor suppressor gene expresses and says, stop, no more, right? We're gonna put the brakes on cell division. So what happens then if I mutate a tumor suppressor gene, which normally turns off cell division? Now it can't. If it normally turns off cell division and I damage it, now it can't stop, okay? So damaging an oncogene and triggering excessive division, or damaging a tumor suppressor gene, right, which usually turns off division, both of them have the same exact outcome, which is uncontrolled cell division, right? Cancer. Do you guys remember we looked at telomeres at the end of the last unit? So we said those were the little pieces of junk DNA where I need to lay down my primers on the lagging strand. And we said that they were junk DNA because once I lay down that final primer, there's no place upstream to lay down a primer and copy the telomere. But it was okay because the telomeres don't code for any genes. So if they didn't get replicated, nothing was lost, right? But what happens each time you replicate DNA? What happens to the telomeres? It gets shorter, right? Because there's always a little piece of DNA I won't be able to copy correctly. So one of the strategies that tumor cells have is the expression of telomerase. So this is the enzyme that adds more telomere. So it adds more junk DNA to the end of the cell's DNA. So normally we said that if a cell ran out of telomeres, what happened to the cell when it was going to replicate its DNA? It couldn't replicate the DNA anymore, right? So cancer cells, because they have telomerase, they can just keep adding telomeres at will. And that means that theoretically they can replicate forever. We call them immortal because as far as we're aware, they can continue to replicate forever because all they have to do is keep adding telomeres and then they can just keep replicating that DNA onwards and onwards. Have you guys heard of, and you should have because you live in the wonderful city of Baltimore, have you heard of HeLa cells? So did anyone make you read the book, although you should have read it because it was enjoyable and fun? Did anybody read The Secret Life of Henrietta Lacks? Okay. HeLa cells are immortal because the mutation that she had allowed them to keep adding telomerases, and that's why they still are in freezers in labs. I mean, we had bajillions of them. Yes?
So did it actually kill some of the cancer, like some of the diseases as well?
Well, so we use HeLa cells to learn if things we've come up with can kill cancer cells. But you would never give anybody HeLa cells because if you injected HeLa cells into somebody, they would just cause cancer. Actually, they're used in mouse models all the time. If you inject HeLa cells into mice, the mice develop cancer, and then you try to use whatever you've created to see if it'll kill the tumors, kill the cancer, or sometimes accidentally kill the mice. It happens. We do our best, but stuff works really well in tissue culture, and then you put it into a living organism, and you're like, oh, interesting. it died. Looks good on paper, not so good in actual organisms. So there are lots of different types of cancers. So maybe you've heard of some of them, and hopefully not because you have personal experience with them, though I suspect some of you do. There's carcinomas. So carcinomas begin in the tissues that line organs. You have sarcomas. We find those in fat and muscle, cartilage, the bone, all the bone cancers are sarcomas. These two, leukemias and lymphomas and myelomas, these tend not to form solid tumors. Instead, these are what we call bloodborne. So you generally don't end up with like a solid tumor like sticking out of the abdomen somewhere. These usually, we find them because your blood counts off in one way or the other. And probably the least survivable of all will be any of the central nervous system cancers. Oops. So anything in the brain or the spinal cord. They tend because they are inside of your brain and inside of your spinal cord. Anytime you have a tumor growing in any of those tissues, what they do is they push back on the healthy tissues. But because they're inside of your skull and your spinal cord, where do the healthy tissues go? They have nowhere to go, right? So the cancerous tissues usually push so hard on the healthy tissues that that's usually what ends up killing the patient. They're no fun because they have nowhere to go, okay? But anywhere where you have a cell in your body, you could potentially develop cancer. These are actually my favorite. These are fairly benign. They're just kind of wacky to look at. See that one right there? That's a pigmented cell in the eye. They call it surfer's eye and farmer's eye sometimes. So if you're not wearing eyeglasses that protect you from UV, like sunglasses that protect you from UV, you can get pigmented cells that become cancerous. They're usually encapsulated, so they just cause a like weird little shape to form on the eyeball. This is an unencapsulated glioma. So the brain actually still looks like the normal brain shape, but that's because where all the tumor tissues are, it's pushing against the normal tissue, and it's creating, in this particular patient, intracranial pressure got so high, that's what actually ended up killing them. This is probably the most common type of cancer in the United States. This is a melanoma. These guys are particularly scary because they are usually 5 to 10 times deeper than they are wide. So by the time you see them, they've already invaded into the bloodstream, and that's why the 5-year survival rate on melanomas is actually fairly low. This one is super easy to see because it's on somebody who has very, very light skin. And I know that people who have darker skin think they're immune to melanoma, but actually they are not, and it's more dangerous because it's harder to see them forming, okay? And last but not least, this is somebody's spleen. This was actually a metastatic. This was metastatic. Spleen cancers are incredibly rare to be primary. This was metastatic. It came from a breast cancer patient. Oh, and the one in the center is leukemia. This is blood cell.
Is the one in the top left white color?
It is not. So they're pigmented cells in the eye. They're just, like I said, they're benign. It's called surfer's eye, or also sometimes farmer's eye. So people who spend a lot of time out in the sunlight without wearing sunglasses, UV light damages the pigmented cells and can cause that little tumor. This one?
No, the one at the bottom.
This is the spleen. So this was a metastasis from breast cancer. Primary splenic cancers are incredibly rare. Almost all of them are metastases from other sites. But it's a common place for the metastasis to show up because it's very rich in blood flow. So all the blood ends up going through the spleen, but remember the metastatic cells were in the blood as well, so they all end up in the spleen, and that's a common site for metastasis.
And you can get cancer in your eye.
Mm-hmm. Like I said, that one's benign though. They're usually encapsulated people who have them just, as long as they're not covering like the retina, it doesn't affect your vision, and it just, like I said, looks kind of weird, but not a big deal.
So can you have it outside like the white?
Yeah, you can have it in the sclera as well.
Can you see it like really?
As long as they don't grow into the retina, you, as the person who has it, generally can't see it at all. And we wouldn't remove it unless it's covering the retina and actually impeding vision. Otherwise, we would just leave it alone because it's actually safer to just let it be than it is to poke around. Hmm? That's the other reason, yeah. Any type of surgery on your eye is miserable. So if it's not bothering you and it's encapsulated, we would just leave it alone. Be like, that's fun fact, leave that there. Wear sunglasses.
Or would it be passed on to the,
If it starts to occlude vision, then we would take it out.
no, I mean, like, can it be passed on?
Mm-mm.
Okay.
Nope. That happens independently each time. Spend too much time in the sunlight. Wear sunglasses.